I just put bleach into the bucket because this is now how I'm going to get rid of, right, these particular plates. And so when you get rid of plates in the bleach, what you're going to do is you want to open them up and slide them into the actual bleach. And so what I'm not doing is I'm not throwing them in, I'm not dropping them from way up here. I'm taking them, I'm putting them in so that the lip goes into the bleach, and then I'm letting it slide in. Okay? And so remember, you want to kill all of these microorganisms, and so concentrated bleach is the way to go. And the reason I bought this particular bucket, well, it was cheap, is that I can do this with a little bit in there, and they're moving around, and they're essentially now they're sunk inside. Okay, so right now they are getting disinfected and completely obliterated with that bleach. Okay, so now what we're going to do is the culturing microbes in broth lab, right, for the aseptic technique. And so what we see for this is I've got a whole lot of additions on this particular lab, right? First lab where you're working with microbes in high numbers. So remember, safety is priority number one. And then it tells you a variety of notes, which I tell you in the video here, but also make sure you tick everything off. Watching a video of me doing this is great, but have this with you when you're working, and so that when you do everything, you're safe. Okay, so I've got my 4 broths, and as you can see up close, I already have them labeled: yeast, E. coli, Staph epi for Staphylococcus epidermidis, and colony X. Okay, so what I want to do first is a part that's not even written in this lab. Okay, so here I'm going to show you how to do it for the very first time, and that is we're going to work with colony X, which is this. Okay, so what you need to do is open up the nutrient broth, very briefly put it in the flame like this. Okay, briefly because this is plastic, and if you keep it in there, it'll melt. Okay, put your lid open side up. There we go. Maybe this will be easier to see there. Okay, and then what I'm doing is I'm taking my loop, I'm sterilizing it just like you did for the previous lab. It's nice and red hot. Okay, make sure you get all of the braided part because this is going to go into this liquid, and so you don't want any microbes on this other than the one you're going to work with. Now, you know you need to wait 10 or 15 seconds, but in this case you can also cool it down a little quickly by putting it in the liquid. And you probably didn't hear that on the microphone, but I heard just like if you put any hot metal in water, right? And so if anything, that'll just warm up the liquid just a little bit, okay? And so now what I can do is I can go in and I can get my colony X. It's taking me a while because holding it so the camera can see it. There we go. I've got colony X. I am now going to put it in the tube. And I'm going to now swirl the loop like this, right, to make sure all of the bacteria come off of the loop. And even if there's still a little bit left off, what you can do then, put this back. First and foremost, sterilize this loop. Right? If it was just wet, so it's going to take a little bit longer. You need to make sure that you don't see anything on there and that it gets red hot like this, right? That's what we want. So all of it, red hot. Because again, what we're doing is we're actually incinerating the bacteria. All right? And now, go ahead and close this up. And often what you want to do is just another quick through. Close this up. And if there's any type of solid material in there, right, then what you can do is close it up tight and then shake it to make it homogeneous, but you need to make sure it's closed up really tight. And I'm not getting too out of hand because if for some reason this were to fail, I would want the liquid to go right here where I could disinfect it. Okay? And so then go ahead and put it over here. And I'm putting it in the back to remind me that I've already done that one, right? The front I need to do, the back I've already done. And so this is finished. So I'm going to take this plate. I don't need it anymore. I've got my photos. I've already entered the information into the data table, and I've taken my colony X. So this is done. So what should I do with this now? That's right. I'm going to go ahead and put it over here in the bucket. And so I'm going to go ahead and put it, make sure it goes in the liquid so that there we go. And so that is now getting disinfected. Okay, so now we've got one. So now we're going to go to kind of the instructions in the lab manual, and we don't need this anymore, the loop. And so now what we're going to do is we're going to take microbes out of our little baggies here, right? In terms of what we've got in this one is we've got our E. coli. And what I suggest you doing so that you don't get them mixed up is just take out one at a time. So here's E. coli. I'm going to work with that. Or what you can do is you can take your E. coli and put it with your E. coli, right? And then you can take your Staphylococcus epidermidis. Actually, look at that. So I grabbed two, thinking, well, one is E. coli and one is Staphylococcus epidermidis. But look at that, I just caught myself, right? You want to pay attention. You may recall when I was showing you how to do the inventory of your lab manual or of your lab kit that I got an extra E. coli culture. So there it is. So I almost goofed, but I didn't. So what I'm going to do is give me just a moment here, and I'll go ahead and grab that other culture. Here we go. Here we have the Staphylococcus epidermidis. And so we are back on track, getting it done. And so I'm putting this Staphylococcus epidermidis with my Staph epi, right? And so this third tube, you can see I followed the directions where I put the amount of water in there and then half a teaspoon of yeast, and it is doing that. So you'll be able to do that with no problem. Okay, so this is, you've got a little tube. This is the first time you're doing it. And it's one of those things where there's a bunch of E. coli in here. You don't want this to go all over the place, right? So what I like to do, the lab manual says put it on a flat surface. Hey, I like to put it in the little thing right there, right? That way, if it's not going to end up getting tilted or falling over, right? It's going to be right there with me to use. So what we're going to do is we're going to need a sterile pipette. Okay. We need to open up our culture broth tube, give it a little quick flame treatment, put this lid out of the way. You don't want to work over your lid because stuff can then fall down into it. So put it out of your way, and I'm just going to put it up here. You can't see it, but I don't think you need to. Okay, I'm going to go ahead and loosen the cap. All right, and so I'm going to take this out. Right, and when I took it out, it's sterile inside. Don't hold it down here. Hold it up near the top. Okay. And so what I'm going to do then is I'm going to show you right here, is I'm going to open this up. And your fingers, so see how I've got, there we go. I'm pulling this tight against my fingers because you don't want there to be like a bunch of glove hanging over the edge. Okay, so you want the tips of your fingers nice and tight like that. Okay, that way there's no extra. Like see how this is kind of baggy up here? You don't want baggy. Avoid the baggy, nice and tight, all right? So what we're going to do is, and while I'm holding this to make sure that none of this touches anything, right? I'm just kind of moving it around. And so what I'm going to do is open it up. I'm going to move this cap out of the way, again, this way right here. The protocol is to take 250 microliters. What I'm going to do is I'm going to fill it up to right about there, or you can fill it up to this particular 500 microliter. So once you bring it up to that level, okay, then go ahead and lift it out of the liquid, and then you can pull it up, and now the liquid is inside there. What you don't want to do is you don't want to get in the habit of trying to get it up to that level and then trying to hold it like this, right? Because if you do that, then stuff can accidentally come out, right, if you're a little bit shaky. So you don't want that to happen, right? So what you do, again, is you go in, you squeeze the pipette before you put it in. You put it in, you pull it up to where you need it, you bring it out of the liquid, and then you just let it come up into the bulb. You wipe the inside so that you get any extra off there. And then what you need to do is take this, put it inside, and go down about halfway, and then slowly release the liquid. Okay? Now the lab manual says to vigorously shake this. Well, let's see if we can get away with not doing that. I'm going to just kind of swirl it around with my tip. And I'm going to go ahead and up, down. All right, so basically I'm mixing it like this. And so if you can do that gently, don't feel in a rush, don't do things hard. I am just doing this. And now I pull it all up. I've got my E. coli solution completely in there. I can put this back down. I then put this into this tube, and I pipette it and release it. And there we go. The E. coli is going in. Okay? And then what I'm going to do is I'm going to take this pipette. Now, what do I do with this pipette? That's right. I'm going to go over to my bucket over here, and I'm going to put it in, and I'm actually going to suck up a full bulb of bleach, right? So that had E. coli in it, and so basically, I'm just going to go up, down, fill it up with bleach, and then leave it in the bleach bucket. That way, like that E. coli right now is, I would say, it's probably all killed right now, right? That's concentrated bleach solution, and the E. coli was well dispersed, so it's completely killed. Okay, and so for your vial as well, one thing I could have done is I could have put bleach into the vial and closed it up and put it in there. And maybe I'll do that for the next one. But for what I'm going to do now is I'm just going to go ahead and put the open vial in there as well as the cap. And I'm getting close enough to the liquid so that it doesn't splash all over the place, right? Okay, so back to the tray here. Then what we want to do is we want to close this up. And there we have it. And so now with the E. coli, what we see is we've got a suspension here, right? We see that there's kind of a lot of flocculent material. What we want to do is, and we'll test it out to begin with, do this really quick, make sure there's no liquid coming out, right? And what we're doing is now we can go ahead and shake it vigorously to try to disperse it a little bit more, okay? Because we don't want it to have big chunks in there, because then it'll look like what it's supposed to look like at the end. So that's done. We put this to the back. Let's do the Staphylococcus epidermidis. It's the same technique, and that's one of the things about this course is that some things you do repetitively, and that's how you gain the skills, and that's how you become a microbiologist. Because I know that you are nursing students, and you're nurses, right? But this is a biology, microbiology class, so I'm treating you like microbiologists. Okay, so what are we going to do? We need to get some of this into there, and then all of this back into there, and then we need to bleach that. Okay, so what we're going to go ahead and do is we can go ahead and open up the vial here. Put the cap right so that the cuppy end is open. We can open up the nutrient broth. Give it a quick flame. Again, don't flame it too much, because if you melt that and you put the cap back on it, when you shake it, stuff will start coming out. So don't get carried away. All right. Again, just grab it by the end, put it in there, squeeze first, put it in, pull it up to about the halfway mark, and bring it up. And to be honest, if you wanted to go ahead and bring it up all the way to the 1 milliliter mark, you could do that, because this vial can handle that amount. And so what we can do is put it in there. I'll hold it up, even though you don't need to, just so that you can see it in the camera. I'm going to go ahead and dispense the liquid. I'm going to put my pipette down, swirl it around, and then I'm going to go up and down gently. If it seems like it's getting caught, don't try to force it. Be gentle, right? And I'm going to go ahead and put this down on the surface because I like it to be stable. I don't want to end up dropping anything, because if you drop liquid, it will splash around, and that's what we do not want. Okay? So if you wanted to do more like the lab manual, that's fine for shaking it. So for example, let's do that. Let's show you an example of another way you can do it a little bit. After you put it in, just like we're doing now, you can put this in like this. Okay? And that's why I didn't want you to hold this way down, because I knew that you can put it in there. So to shake it like the lab manual says, right? Shake it like the lab manual. There we go. And again, I'm doing it over the tray. I'm doing it low to the tray, right? I'm not doing it like up here, right? All around where I'm shaking it like this, right? No, don't do that, because all of a sudden you've got the bacteria going all over the place, right? No, keep it down at your workspace. Very nice. Safety first. right? You're present when you're doing these labs. You're not just trying to get through them, you're being present. Okay, so now when you open them, the thing about there being liquid in there is that you just need to make sure that when you pull the cap off, you do this and see how there's like little bubbles, okay? So that's one reason why I don't like shaking it sometimes. What I'm going to do is I'm going to go ahead and take this cap and I'm immediately going to go ahead and put it into the bleach bucket, right? And then we can go back, and down here we need a pipette that doesn't have liquid in it, empty pipette, and then squeeze it. And since there's bubbles on top of this, so if I just stick this in, the liquid is going to come out and go over and contaminate it. So what I'm going to do you see that bubble right there? I'm going to go ahead and squeeze when I touch it. Squeeze. There we go. And now I've just sucked up that bubble worth of stuff. And then I'm going to get the rest of the bacteria. All right. I'm going to go ahead and put it in my broth tube. There we go. And then what am I going to do with this tube? I'm going to go ahead and bring it over to the bleach bucket. I'm going to go ahead and suck it up, release it, suck it up, right? Fill it up nice with bleach, and then I'm going to leave that in the bleach bucket. And I've got this kind of at a weird angle over here just so that I can work on this, you know, and I want to be in front of the camera seeing you guys. So definitely have the bleach bucket somewhere where you can work on it nice and easily. Okay, so back to the tray here then. What we're doing is, oh, that's right, we need to go ahead and get rid of this culture vial. So I'll go ahead and stick it in the bleach. There you go, die, 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 right? I mean, I love the bacteria, but we need to kill them at the end. Okay, so back to the tray here. Then what we need to do is go ahead and a quick flame, cap it up, do a little bit of inversion, make sure that there's no liquid coming out, and then you can do a little bit more vigorous shaking to try to make it homogeneous. All right. So the last one that we're doing then is the yeast. And so the yeast, I've had this going now for 15 to 20 minutes. And so what you've done before is still what we're doing. Go ahead and flame it gently. We take a new sterile pipette. Go ahead and just pull just right to about that mark. You see how basically what we're looking at is to this mark right here. You don't want a huge amount, so right there, at the bottom where it bulbs out. And then what I'm going to do is just go over, take it, and put it in. Okay. All right, now since this was just baker's yeast, I mean, I can just throw this in the garbage, right? I mean, there's no problem with this. It's not like it's, you know, E. coli or Staphylococcus epidermidis. No, the answer is you treat everything the same way. Even though that's baker's yeast that you would eat because it's in, well, bread, right? We need to treat it as the microbe that it is. This is a microbiology class, so we never cut corners. So we're going to go ahead and bleach it just like we did for Escherichia coli and for Staphylococcus epidermidis. Bring it up, let it go, fill up the bulb, right? And then go ahead and leave it in there to stew and to be destroyed. Okay, so back down to the tray then, what we're doing is we give it the heat treatment, very gentle, close it up. We first invert it gently to make sure it's not leaking, and then we shake somewhat vigorously. Oops. All right. So now I did have a student one time melt this by accident when they were doing it. So if that's the case, if you can still get the lid on, just don't shake it, right? If you think you melted it, then don't invert it because you'll get leakage. Just say, well, okay, I'm just going to go ahead and have to leave that as. Just tighten it up. Okay. So now we have our 4 prepared cultures that we're going to go ahead and incubate in our chamber, right, for 48 hours. And I'm not going to take my lid and put this on right now because I want the outside of this to not be contaminated. Now again, do I think I just contaminated my gloves? No, I think I did a good job. However, we still, when we have gloves on, we don't touch anything that you're going to have elsewhere. And so the inside of this can have the microbes, but I'm going to wait until after I wash up before I go ahead and put this on it, close it up, and bring it away. Okay, so what we're going to do is we're going to go ahead and leave this for 48 hours tucked away. And then what I'm going to do right now is go ahead and stop my flame, pull this out, And what do we do for the inside of the tray here? All right, we go ahead and we grab a paper towel, and what I'm doing is I'm grabbing it with my glove, but I'm holding it with my arm. Again, I don't want to be grabbing things with these gloves. Get used to that, right? And then I'm going to go ahead and now this Clorox bleach, this is for my microbiology lab. So I'm not going to stick this underneath underneath the sink for anybody in my house to use. No, this goes with my microbiology stuff in the cabinet that I have everything up because I'm handling this with my gloves. So you don't need to be paranoid about this class, but you need to be very purposeful in how you act in the activities you engage in. So what I'm not going to do is I'm not going to start spraying this and have stuff all over the place. What I'm going to do is I'm going to use my paper towel, and I'm going to use that as a barrier. Okay? And so what we have here then, yep, and I certainly don't want to knock over anything. Let me do this again. Let me go kind of around, just like we do our lab bench here at school, right? And so then we can go ahead and make sure we wipe this quite well. Lots of bleach. And we don't rinse it because we want that bleach to stay on there and kill anything that's there. And I was just touching the edges with my gloves, so I'm wiping that off. Right? That's why I like these trays instead of just doing it wherever. Okay? So now, so what do I do with this? Right? So one thing I'm going to do with this is, well, I just handled this. I'm going to put this in my bleach too. Okay? All of this stuff that has been in the tray that you've been dealing with should go in the bleach bucket. And so in terms of the yeast, right, that is something where, okay, Dr. Willett, which is it going to be? Are we treating it like the microbes across the board, or do we say, look, it's just the yeast? Yep, it's going in here. And this concentrated bleach can take that a little bit of liquid that I've got in there because it is concentrated bleach. If we were to put a whole bunch of water in there, then that would be problematic, okay? And so sometimes what people like to do is they say, well, I've been handling all this. I'm just taking all of this, and I'm putting it in the bleach, right? I've got another packet of yeast. I was handling it with my hand. I'm putting it in the bleach. When in doubt, bleach it out, right? And so then when I look at this, I'm just going to go ahead and push a few things kind of around, make sure that everything looks good. I'm going to use this cup of the yeast, and I'm going to go ahead, because I didn't fill this up with bleach. I mean, I didn't want to put like, a gallon in here, but for example, this is bleach, and I'm just pouring it on my Petri dishes to make sure that everything is dying. And yes, I am getting a face full of bleach right now, so depending on how sensitive you are, you may want to wear your mask or just do it some other way. And so then basically everything is covered. All of my plates are submerged, right? And so I'm going to leave this now for 15 to 20 minutes before I do the next step, okay? And so let's wait that 15 to 20 minutes. All right, I've waited 20 minutes, and so what I'm going to do is I'm going to go ahead and start fishing some of this stuff out of here. Be careful to not get my hands contaminated. And so as the lab manual says, it says to wrap things up in paper towels. So what I'm going to do is I'm going to go ahead and put in a bunch of paper towels, right, which will help me soak all that up. I'm going to grab the material. And what I'm going to do is I'm going to leave my left hand, not in the bucket, okay? So what I'm going to do is I'm going to take this material, it's a little messy, but that's why I've got the bucket, right? The bleach, remember, the bleach is your friend. It's killed all the materials, okay, all the bacteria. So this is where I could use an assistant as well, but that's all right. As long as I don't ever touch that with my gloves, right? I'm good to go. And so let's go ahead and grab these Petri dishes out of here. Yep. And this is a nice sturdy bag, right? This is not some cheapy bag. This is a nice sturdy bag, okay? And so I've got all my materials in there. And what I'm going to do is use the zipper on this. Put this in another bag. Right, and at this point, I mean, you use bleach on this, so everything should be killed. So I'm not sitting here worried like, oh my goodness, there could be pathogens in here. What I'm worried about is getting bleach everywhere, right? And then what I'm going to do is take this, and go ahead and take my gloves, bring them inside out, put them in, grab another paper towel, dry my hands off, because as you know, when they've been in gloves, they can get rather moist. And so this right here, I'm going to take this, I'm going to put this in another garbage bag, like a black heavy-duty garbage bag, and then I'm going to put it directly into my garbage can out in my garage. I'm not going to stick this inside, right? This is now to go away, get out of my house, everything's dead. I've got a little bit of bleach in here left, so I'm going to go ahead and I'm going to make sure I swirl it around, right, to make sure that anything on the edges. And you have two options for this. You can either lay down a whole bunch of paper towels in there to suck it up and do just what I did. I don't like to have all that much in one. I like to do it in discrete. Or you can go ahead and pour it down essentially into your toilet and then clean your toilet afterwards. So either way, the lab manual recommends that you take a whole bunch of paper towels and put it down there, but I'll leave that up to you, right? So that's how we, now that I've got my gloves off, right, I can go ahead and seal this up, and I will then incubate this for 48 hours. I've got my tray over here. I've already cleaned that up. It's all squared away. I can go ahead and put this away, and I'll go ahead and put everything away. In my case, it's this bench that I keep everything in. In your case, hopefully you've got a cupboard somewhere, you've got a chest, a place where you're, you know, if you've got a Marcel running around, it doesn't get into it. And so I'll see you next time to see the results from this incubation.